So today's lecture will be the lecture 2 on bacteriophage lambda. So yesterday towards the end of my lecture, I told you in lytic pathway the genes are expressed sequentially. There are 3 stages. So you have immediate early, you have delayed early, and you have late. And we have seen how these genes are located on the genome. We have seen the diagram also. And in the center of the genome, you have a regulatory region. That regulatory region has been expanded here. This is the regulatory region. You have this CI or CL gene, which codes for a repressor protein. Then you have a promoter PM. M stands for maintenance. Then you have one more promoter site PR, because it is on the right hand side, we call it as PR. And within the promoter, as we have been seeing, there is an operator region OR on the right hand side. Then there is a gene CRO, which codes for an anti-repressor protein. Then there are a couple of transcription termination sites. And then there is C2, which is a positive regulator protein. Okay. So this is the regulatory region, which is in the center of the genome that has been magnified for you. On the left hand side, you have a promoter which is called as PL. And within the promoter, there is an operator region which is called as OL. And then there is a gene called N, which codes for an anti-terminator protein. And then you have another transcription termination site, and this is C3. C3, which is also a positive regulator protein. So let us first see as to how lytic pathway proceeds. So bacteriophage infects the bacterium. Infection is the first stage. Okay, it will get attached to the bacterial cell surface and then will inject the DNA into the bacterial cell. Okay, DNA will be injected into the bacterial cell. Please remember that bacteriophage lambda doesn't have any resources apart from these limited number of genes. It doesn't carry any enzymatic apparatus or it has any resources. So it utilizes the host machinery, the bacterial cell machinery, the enzymatic system, as well as the resources of the bacteria host. All right. So in lytic pathway, what happens as soon as the DNA gets into the bacterial cell? So this is the DNA, bacterial DNA. We have seen earlier, we have few genes on this side. We have some other genes on this side. Only the regulatory region has been expanded here. So on this side, you have genes for replication, then you have an anti-terminator protein Q, and then you have genes for lysis. On this side, you have recombination genes, you have genes which code for tail proteins, and then you have genes coding for the head protein. So as soon as this DNA gets into the bacterial cell, what happens? The bacterial RNA polymerase, they will bind here. So bacterial RNA polymerase will bind here. This is the promoter region. So now you are very well aware that promoter is the site where bacterial RNA polymerase comes and bind. So there are two promoters and the arrows, they indicate their orientation. So bacterial RNA polymerase will bind to promoter on the right hand side calling PR, and RNA polymerase will bind to promoter on the left hand side, we are calling it as PL. And after binding, they will start transcribing these genes. The first two genes to be transcribed will be CRO. So this is CRO mRNA and this is nmRNA. CRO is an anti-repressor. It will be translated and you will get this protein. This is an anti-repressor protein. And N, is an anti-terminator protein. All right, these are polypeptides. And this is this sign of a polypeptide chain or a protein chain, right? So anti-terminator protein, what does it do? See, you have this transcription termination site. In the absence of this anti-terminator protein, the RNA polymerase which has started from this promoter would have stopped here. Okay, but since anti-terminator protein is there, it allows RNA polymerase to read through these transcription termination sites. Please remember, I am repeating once again, there are transcription termination site. These are transcription termination site 1 and this is transcription termination site 2, T1 and T2. So RNA polymerase otherwise would have stopped here. But since in the first phase, immediate early phase, you have these two genes transcribed, one of them codes for an anti-terminator protein. This anti-terminator protein allows RNA polymerase to get past these transcription termination site and it transcribes the C2 protein. On this side also, this N, there is a transcription termination site. Because of the anti-terminator protein, RNA polymerase will read through this transcription termination site and will get into C3. These two genes will be transcribed in the next phase. They are the part of the delayed early genes. So you have C2 and C3 positive regulator proteins, you have recombination proteins, you have replication proteins, right? And you have Q. There are four sets of genes which are transcribed in the next. So you have here, you can say positive regulators C2 and C3. Then you have recombination genes, 7 genes. You have replication, there are 2 genes, and there is Q. These are the 4 sets of genes which are transcribed in the second. Akash Thakre is now joining. Second set delayed early. Okay. So because of the presence of anti-terminator protein N, RNA polymerase will not stop here. RNA polymerase will get into C2, will transcribe C3, and it will also go beyond these. On this side, you have, genes which are for replication, and there is a Q here. And there are 2 genes here for replication. So all those will be transcribed. And once Q has been transcribed, it also codes for an anti-terminator protein. So this protein, Q protein, it will allow RNA polymerase to read through whatever other transcription termination sites may be there. All those sites on this side, they will be, RNA polymerase will read through them. So what do you have? You have on this side, you have those 2 genes. S and R, which codes for the proteins which are required for lysis. So now you can understand how sequential expression. In the first phase, only 2, N and CRO. And the direction you can understand, RNA polymerase, this is the bacterial RNA polymerase, it will bind to the promoter and then will start transcribing. In presence of N, whatever transcription termination signals are there, RNA polymerase will read through them and will get into the delayed early genes. And there are 4 sets of genes. You have 2 positive regulator genes, you have recombination genes, 7 of them, you have replication genes, and you have a Q. All these will be expressed sequentially. In presence of Q, which is also an anti-terminator protein, RNA polymerase will read through the other transcription termination signals and will enter into the late genes. And there are 3 sets of genes in the late region. You have genes which code for tail proteins, 11 of them. They will be transcribed and tail proteins will be synthesized. You have 10 genes which code for head proteins. And finally, there are 2 genes which code for proteins which are required for lysis. So they will be expressed. Okay. So the timing is very critical. Genes which code for proteins required for lysis, they will be expressed only when the phage particles have been completely synthesized and the DNA has been properly packaged. Okay. Premature lysis of the bacteria will result in what? It will result in liberation of freeing of half-formed progeny particles. Half-formed. and they will not be able to survive. So it is very important that these genes, these two genes are expressed only at an appropriate time when the DNA has been replicated. Not only DNA has been replicated, but it has been properly packaged into the code proteins. Okay, and this timing is very crucial, and this is achieved all the time. There's a lot of precision in that. These two genes will be expressed and the proteins will be synthesized, and these two proteins, they will be responsible for lysis of the bacteria. Okay, this is pretty simple, very straightforward mechanism how lytic pathway proceeds in bacteriophage lambda. Okay, I'm repeating it once again. The bacteriophage lambda will infect the bacterium, will insert the DNA into the cell, bacterial cell, and once the DNA is there, this is the DNA. We have this central portion, the regulated region expanded. Rest of the DNA is on this side, and rest of the DNA, remaining DNA is on this side. Okay. The bacterial RNA polymerase will bind to these promoters. You have one promoter on the right hand side with the orientation shown here, and you have another promoter in the left hand side with the orientation shown over here, right? And RNA polymerase will start transcribing the bacterial DNA. So first two genes to be transcribed are CRO and N. CRO on the right hand and N on the left hand. And N is an anti-terminator. In presence of anti-terminator protein, RNA polymerase will read through the transcription termination sites which are there in between the immediate early genes and the delayed early genes. And one of the genes which is expressed in the delayed early is a Q, which also codes for an anti-terminator protein. This anti-terminator protein Q, it will be responsible for the RNA. It will make RNA polymerase walk through the transcription termination signals which are present between the delayed early and the late genes. So it will get into the late zone, late segment, where you have genes for tail proteins, you have genes for head proteins, and finally, in the end, these two genes for lysis, they will be transcribed, proteins will be synthesized, and bacterial cell will be lysed, and about 100 odd progeny particles, they will be liberated into the environment. Okay, and normally, as the logic says, that lytic pathway will occur when the conditions are favorable, when you have ample of nutrient supply, when you have environmental conditions favorable, and then only there is a point, liberating the progeny into the environment. Okay, this is a logic says. I told you what decides whether the bacteriophage lambda will go for lytic pathway or lysogenic pathway. There are two possible assumptions. One is the conditions, and the second is the genotype of the bacterial cell. It also depends on that. We still don't have a very clear idea as to what is the trigger or what decides ultimately bacteriophage lambda will go for lytic pathway or bacteriophage lambda will go for lysogenic pathway. Lytic pathway is pretty straightforward, doesn't end over here. It is interlocked with the lysogenic pathway. So we'll come back to this lytic pathway in the later part of the lecture. Now let us understand the lysogenic pathway. Okay, lysogenic pathway. The first two events remain the same, that the bacteriophage will infect the bacterium, infection, and insertion of DNA into the cell. Into bacterial cell. Okay. After that, what happens? Lysogenic pathway, for the sake of understanding, it can be divided into 3 stages. It is a little bit more complicated. So let us understand what is stage 1. Stage 1 is called as establishment of lysogeny. Or establishment phase. Second is maintenance. And the third stage is termination. And as usual, Establishment is probably the most difficult of these three stages. Okay. Once established through maintenance circuit, lysogeny will be maintained, and I have been telling you that bacteriophage can remain in the bacteria, can remain with the host for practically infinite period of time. So lysogeny can be maintained for generations. And as soon as it detects, the bacteriophage detects that the host has become inhospitable, the host is no longer a safe haven, at that time the lysogeny will be terminated and the bacteriophage will switch over to the lytic pathway. So we'll understand these three stages one by one. Okay, so let us first understand what is meant by establishment of lysogeny. Okay, so I told you, if you see the previous diagram, that there is one more promoter here, which is called as P on the right hand side and E. Promoter R means on the right hand side, and there is one more E over there. It is for establishment. And see the orientation of this promoter. The orientation of promoter is this. Okay. Is it too complicated? Should I draw it once again? Okay, let me draw it once again. So you have CI in the middle, CL or CI. Then you have a promoter maintenance. You have PROR. Then you have CRO over here. Then there are a couple of transcription termination sites. Then there is a C2. And then there is a promoter called PRE. PRE. On this side, you have PLOL. You have gene N. There is a transcription termination signal here. Then you have C3, right? Now in lysogeny establishment, once the DNA, and then you have rest of the DNA, we are right now not talking about. Please remember, in lytic pathway, except for this gene CI or CL, I don't know whether it's CL, except this gene, all other genes were expressed. Barring this, all viral functions were expressed in lytic pathway. All other genes were getting expressed except for this. Whereas in case of lysogenic pathway, only this gene gets expressed, all other viral functions are temporarily suspended. Okay, they are not lost, they're temporarily suspended. As I told you, as and when bacteriophage will realize that the host is no longer a safe haven, it will switch over from lysogeny to lytic pathway. That means all those viral functions will be restored. So they are suspended, they are temporarily suspended, they are not lost. As and when it requires, those genes will be expressed again. Okay, so you have the bacterial DNA into the, you have the bacteriophage DNA into the cell. And now for lysogeny establishment, I told you, right now we do not have exact idea as to what decides whether the bacteriophage will go for lysogeny or the bacteriophage will go for lytic pathway. I told you there are two possible assumptions. One is the environmental and the nutrient supply condition, and the second is the bacterial genotype. Okay, but in lysogeny, what happens? So you have a promoter here, and the orientation of this promoter is, please remember, the orientation of promoter is this. Orientation of PR is this, and orientation of PL is this, right? RNA polymerase will bind here, the host RNA polymerase. Host RNA polymerase will bind here and will start transcribing this DNA in this direction. So this CL, it will be transcribed. You have this CL RNA, which will be translated immediately into this is called as repressor protein. repressor protein. So this repressor protein will be formed, okay, and this repressor protein, it is a monomer of 26 kilodalton. The active form is a dimer. Is a dimer of identical subunits. So you have two subunits of 26 kilodalton each in this repressor protein. It is a dumbbell shaped protein. You know, it looks like this, dumbbell shape. In the gym, you have these dumbbells. Okay, so this is the N terminus. This is the C terminus. From amino acid number 1 to 92, this is N terminus. This is also called as DNA binding domain. And there is a central rod region, and then C terminus, you know, it starts from 1112 to 132 amino acids. This is also called as dimer binding domain. Dimer binding domain. Okay, so what does it mean? It means that the two subunits, they will be joined to each other with the help of this C terminus domain, dimer binding domain, whereas this N terminus domain, which is a DNA binding domain, this repressor protein is capable of binding to the DNA. Okay, so this dumbbell shaped repressor protein of bacteriophage lambda, you know what is the site of repressor binding? The site of repressor binding is the operator region. Operator region. All along we have seen in case of lycopyron, in case of trypopyron, that repressor protein binds to the operator region. So this repressor, first few molecules of repressor, those which are synthesized by RNA polymerase, which has originated from promoter RE, promoter on the right hand side, establishment. Okay, those first few molecules of repressor, they will go and occupy. So let me draw it for you here, the operator region. They will go and occupy the operator region.